Hi, welcome to our channel BioWell. Today our topic is malarial fever which is caused by Plasmodium species. Do you know one thing? In India, around 3,50,000 people were suffered by malarial fever and every year. So, just to see how the Plasmodium causes malarial fever in our human body. Just let's do our topic. Okay, when you hear the Plasmodium, that time you should remember the three letters. That one is P, S, H. Just you see P, the first letter starts with the Plasmodium P. Yes, the letter is here also. Then H, just you remember the hospital P, S, H. Okay, so first the Plasmodium that belongs to the phylum Protozoa. That one is Protozoa. Here Plasmodium is coming under unicellular eukaryotes. First, you should know what is unicellular eukaryotes. Unicellular eukaryotes are nothing but they are single cellular organisms that are having a definite nucleus, mitochondria, and all other microorganelles. Then you see the class Sporozoa. The Plasmodium coming under Sporozoa. Sporozoa means that do not have any cilia and flagella for migration, but they possess the gliding movement. Just you see here, gliding movement means like this. It moves like this. Then you see the order Hemosporidia. From the name itself, you see here, Hem means that one is the blood. It spends some of their life cycle in the blood cells of the vertebrate. That one is nothing but human beings. You see in the first picture, it denotes a slender shaped plasmodium. The second picture, you see structure of sporocyte. This one is very important. Which one is the infective stage of malarial fever? That one is the sporozoid stage. Don't forget, the infective stage of malarial fever is sporozoid stage. You look at this picture. Here, is having the definite nucleus, a definite mitochondria, a definite, you remaining all other organelles are very clear. Okay. So, the next one is the history. When you take the malarial fever, the history is very important, and also two important persons. One is Charles Lavren, who was the first person to notice Plasmodium in the year 1880. You see, Charles Lavren was the French military surgeon. Okay, don't forget. Charles Lavren, the first person to notice Plasmodium in the year 1880. You see, the second person, and that person was Sir Ronald Ross. He was the first person experimentally proved the mode of transmission of this malarial parasite. That one is in the year 1889. For this purpose, he got Nobel Prize in the year 1902. You see, next life cycle of Plasmodium. Plasmodium, when you hear the name Plasmodium, that one is the intracellular sporozoan blood parasite. What is the difference between intra and intercellular? Intra means, you look at this picture, the intra means it is present inside the cell. Inter means it is present in between the cells. Don't forget, okay? Then for fulfill its life cycle, the Plasmodium that needs two hosts. One is man and the one is female Anopheles mosquito. That one is blood sucking invertebrate. The next one is mode of reproduction. This Plasmodium that also two types of reproduction. One is asexual reproduction and another one is sexual reproduction. Here, asexual reproduction is otherwise known as schizogony that occurs in the human body. But in the case of sexual reproduction that occurs in the female Anopheles mosquito, that name is porogony. Don't forget the name terminology. That one is schizogony means asexual reproduction. Porogony means that one is the sexual reproduction. Here, in the man is considered as the intermediate host. What do you mean by intermediate host? Because intermediate host is one of the very important place. Here, this host only gives the support for the growth of an immature parasite into mature parasite. This Plasmodium causes severe defects. That one is effects in the case in human beings only. Then, in the case of female Anopheles mosquito, the sexual reproduction happens in the female Anopheles mosquito. Then don't forget Charles Laveran, who was the first person to notice plasmodium in the year 1880. Sir Reynold Ross, he was the first person to experimentally prove the mode of transmission. For this purpose, he got Nobel Prize in the year 1902. Okay. The next one is life cycle in man. When you take the man, here the life cycle that took place in two stages. One is exoerythrocytic cycle and another one is endoerythrocytic cycle. Here exoerythrocytic cycle that happens in the liver cells. Endoerythrocytic, endo means it is present inside the cell. Okay, the first one is the exoerythrocytic cycle. This one is very important. You see, when the infected female Anopheles mosquito bites a person, it releases thousands of slender sickle-shaped nucleated sporozoids are injected in the blood of the particular person. I already told sporozoids are the infective stage of malarial fever. Then you see these sporozoids are supplied in the blood for first 30 minutes and then penetrate into the liver cells. You see, that stage is known as cryptozoids. These cryptozoids continuously feed on the liver cells and increase in size. That stage is known as cryptoschizon. Don't forget cryptoschizon. Then this cryptoschizon, due to the heavy load, it's going to asexual reproduction. That one is nothing but schizogony. During under asexual reproduction, this cryptoschizon are continuously divided into cryptomerozoin. So you see, in the exoerythrocytic cycle, all the stages that started with C. You see, cryptozoids are continuously feed on the liver cells and increase in size. That stage is known as cryptoschizon. Once it reaches the cryptoschizon, then it undergoes asexual reproduction and it releases large amount of cryptomerozoids. These cryptomerozoids are cryptomerozoids that invade the new RBC cells and some cryptomerozoids that invade the fresh liver cells for further development. So the whole this exoerythrocytic cycle that requires 7 to 17 days for its development. So this stage is known as incubation period. So exoerythrocytic cycle, that stage is known as incubation period. Don't forget cryptozoid, cryptoschizon, cryptomerozoid, that coming under exoerythrocytic cycle. So the next one is endoerythrocytic cycle. You see, the cryptomerozoids, when the cryptomerozoids enter the RBC cells, it feeds continuously its contents and reach the amoeboid stage. When it reaches the amoeboid stage, that stage is known as trophozoids. Don't forget, that stage is known as trophozoid. In the trophozoid stage, it develops a back wall. This back wall increases in size. So when it increases in size, you see here the nucleus is pushed to one side. So that stage is known as signet ring stage. That stage is known as signet ring stage. Then this amoebula stage that occupies the entire RBC. So the RBC becomes increased in size. That stage is known as schizon. That stage is known as schizon. During this schizon stage, the nucleus breaks into 6 to 24 small bits. Each small bits are covered by cytoplasm. That stage is known as merozoid. That is known as merozoid. So first one is trophozoid. Then trophozoid enter in the signet ring stage. Then next, after signet ring stage, it breaks into number of bits, small bits, that is known as merozoids. Then the merozoids try to structure the cell wall of the RBC. So when it ruptures the cell wall, it releases a chemical substance. That one is a toxic substance that is known as chemozoin. This chemozoin is responsible for malarial fever. Don't forget, chemozoin is responsible for malarial fever. Then after that, the merozoids, some merozoids that enter into new RBC cells, and some one is, instead of developing into trophozoid, it develops into gametocyte. So don't forget, for the endoerythrocytic cycle, cryptomerozoids are changes into trophozoid. Then it enters the signet ring stage. then it once it reaches the signet ring stage, then you see the amygdala occupies the entire RBC, so that stage is known as schizon. During the schizon stage, the nucleus breaks into 6 to 24 bits. That one is developed into merozoid. Then the merozoid ruptured the wall of the RBC. Then it released a toxic substance known as chemozoin. That one is responsible for malarial fever. Some merozoids that enter into new RBC, and some merozoids, instead of developing into trophozoite, it develops into gametocyte. So the next one is life cycle in female Anopheles mosquito. When the Anopheles mosquito bites the infected person, you see the gametocyte that enter into the female Anopheles mosquito, when it reaches the gut, this gametosite undergoes gametogony. Gametogony is nothing but the development of gametes from gametocyte. There are two types of gametes. One is macrogametocyte, another one is microgametocyte. Macrogametocytes are nothing but female, and microgametocytes are male gametocytes. Just to look at this picture, in the macrogametocyte, the nucleus is pushed in one side and the cytoplasm is occupied the whole cells. But in the case of microgametocyte, the nucleus is scattered and also the cytoplasm is scattered. So the next one is the next stage is fusion. The fusion of male and female, that one is macrogametocyte and microgametocyte to form the nuclear fusion happens. Nuclear fusion is nothing but fusion of male and female gametocyte. That forms the zygote. Once the nuclear fusion takes place, then only zygote formation happens. Then the zygote is transformed into ookinete. Ookinete is a stage of elongated and movable form. Ookinete is the stage of elongated and movable form. Then the ookinete is changes into oocyst. Oocyst is nothing but the zygote, that one is enclosed in a cyst wall. The zygote is enclosed in a cyst wall. Inside the cyst wall, the sporozoids are developed. So the sporozoids are developed inside the cyst wall. Once it reaches the full maturation, then the sporozoids are try to enter the salivary gland. When the female Anopheles mosquito bites the healthy person, the sporozoids enters into the healthy person and cause malaria. And further, the whole process taken inside the human being. Now I trust you understand. You can understand Plasmodium, life cycle of Plasmodium. Thank you so much.